Our next session is going to be on PyMOL, and specifically we'll go into molecular visualization using PyMOL. We'll also spend a little bit more time talking about the protein data bank. And so hopefully y'all are hanging in there. It's you know, this is a kind of a marathon. We try to struggle between keeping it short so you guys can get back home and back into the lab, but also putting as much information into these sessions as we can. And so hopefully you're able to get up and stretch, get out. Maybe, you know, it's not terribly sunny, so you can't really get out the sun. But we're almost done through this day. And the last day is a short day, so it won't be so bad. So stick with it. Hopefully you got good notes. You know, we're getting good video. And so you'll be able to review things later on if you need to go over stuff. Anyway, so yesterday, and it's come up a couple of times throughout the sessions, we introduced this idea of the protein data bank, which is the database that holds or collects all of the structural information for biomolecules. And even though it's called the protein data bank, you can still find nucleic acid structures there. And so Chris also mentioned the PDB in his talk. And so the protein data bank really has all sorts of types of biomolecules in there. It's really, you could almost think of it as the biomolecular structure database. There's two main types of files that end up in the PDB. We'll talk primarily about protein crystal structures in today's session. But as techniques develop, as people sort of use different types of methods to solve structures, you're seeing more and more types of data in the protein data bank. And so the main two are protein crystal structures, or just crystallography, X-ray crystallography, and NMR structures. And so historically, those have been the dominant ones. Honestly, crystal structures are by far and away the most populous or the most numerous structures in the protein data bank. They outnumber NMR structures, I think, by about 10 to 1. However, in the last couple of years, really in the last 5 years, cryo-electron microscopy has grown as a new and emerging method for solving structures, particularly of large complexes. And you know, we're all here in the age of COVID. All of these structures that you guys have seen, where they have these beautiful sort of large complexes of, you know, the spike protein or different cell surface components, you know, a lot of those are actually determined by cryo-electron microscopy. And so each of these types of techniques you can spend years and years mastering. And so we're not going to be able to cover all the details about X-ray or NMR or cryo-EM in this, you know, hour and 20-minute session. On the other hand, we are going to try to give you enough details so that you can look at a structure, particularly a crystal structure, and assess, is this structure trustworthy? Can I use this for super high resolution work? Should I maybe have a you know, caveat emptor, buyer beware type of a response to this structure? And in particular, a very common problem that we all face as biomolecular scientists is you'll often search something in the PDB and you'll end up with 10 or 12 different structures. And so the question is, well, which one do you use? And so being able to understand a little bit about how the molecules or the structures are determined can give you some ability to determine whether your structure that you're choosing is trustworthy or is the best choice. And so so thinking about the total number of structures, I don't know that I updated this year. This seems a little small. I want to say we're up around 170,000 now. But this kind of gives you that ratio. If there's 140,000 total structures, you know, 126 are going to be crystal structures. NMR is going to be the vast majority of the remainder. And then cryo-EM, you're probably in the thousands of structures, but not quite up to tens of thousands. I haven't exhaustively searched that yet. So Matt is probably the authority in this session on building X-ray crystallography structures. And so we're not going to talk too much about the method. But what I want you to understand is we had that session on model building and the agreement between the model and the data. even though you can go on the PDB and download coordinates for all sorts of different types of biomolecules, what you got to understand is that that's a model too, right? There's experimental data, and you're basically refining your model to maximize the agreement between the model, which is those three-dimensional coordinates that you're looking at, and the experimental data, which in many cases is just a whole bunch of reflections that you pick up on a piece of film or some kind of optical detector. With NMR, it's even a little bit more sketchy how you make this, because NMR, you're not really even directly measuring atomic distances often. You're measuring peak intensities that can be sort of correlated with atomic distances. And so thinking about the X-ray structure and sort of how it's built, generally when you look at an X-ray structure, you get this sort of page of spots. And I didn't even include a slide here because it's essentially meaningless unless you know what you're looking for. But that page of spots, that page of spots with different intensities, and some spots are light and some spots are dark, can be roughly calculated into what's called electron density. And so this is kind of a demonstration of the structure building process where you can see that in the early stages, these dark lines represent your model, and the sort of mesh that's drawn over here sort of represents the experimental data. And the challenge with X-ray crystallography is that in addition to the fact that you're trying to maximize the agreement of the model and the data look different depending on what model you use. And so you're actually using some of the experimental data and then some of the model, and then that's what's used to generate the electron density, and then you can graph that on top of some atoms like we're doing here, and as you refine it more and more, you end up with something that looks a little bit more realistic, a little bit more believable, where, for example, in this guanine residue, you can see that, okay, no, I can believe that, okay, there's an atom here, there's an atom here, and these look like two rings, and this looks like a ribose sugar and the nucleoside backbone. And so, generally speaking, when you're building an X-ray structure, there's actually a lot of calculation, a lot of computation that goes into that. And like I said, we're not going to cover the details of it, but it does lead to the importance of assessing whether that model is good. And so in the model fitting section, we talked about chi-squared. In the X-ray world, they've developed something that's similar, but in this case, it's called the R value. And you can see that as the R value gets smaller and smaller, just like as chi-squared gets smaller and smaller, the agreement between the calculated electron density and the three-dimensional atomic coordinates gets better and better. And so if we think about what we can look for as we look at an X-ray structure on the PDB, we can sort of come up with a laundry list of good things to think about, things that are demonstrative of a good X-ray structure. And so first of all, that R value that we just talked about, that R factor, if it's less than 25%, you can almost think of that as like a 25% error. This is assessing the agreement quantitatively between the calculated positions and the positions determined from the experimental data. Often you'll see R values represented in decimal terms, so this is just 25% or 0.25. They also have, because of this property that X-ray structures are actually using the coordinates and the experimental data to come up with an initial guess, they have something called an R-free. And what the idea here is that the very beginning of your experimental refinement, you take some of your experimental data and you put it in a box, a lockbox, as Al Gore would say. And you would say, okay, I'm not going to look at that data until I'm done with the rest of my refinement. And what you would do is then go and take the rest of the 75% of the data, or 20% or 80% of the data, whatever data that's remaining, and you would use that to determine the structure. So it might not be quite as good of a structure if you had used all of the data to begin with, but that gives you a sanity check at the end of the experiment, because then you can go back and you can compare your data or your experimental model, and you say, how consistent is this experimental model with this data that I left out and didn't use at all in my calculations. And if they're consistent, that's good. That means that your model is actually pretty good. You didn't fool yourself when you were generating your initial estimate of the structure. And so that's what's meant by R-free. Because you're leaving out data from that initial, putting it in a lockbox, the R-free is generally always going to be bigger than the just regular R factor. But you want that to be 25% or less as well. You have a little bit of more leeway. If you're less than 30%, you're probably okay. More than that, I would start to raise my eyebrow at that particular structure. Resolution, we talked about this on the first day of the session. Generally lower, and the smaller the value of the resolution corresponds to higher resolution. So if I say higher resolution, the value is actually lower. So 2.5 is pretty good. 1.5 is better if you can get it. And I would say 1.5 angstroms resolution is higher resolution structure than 2.5. So again, sorry for the nomenclature. As you get lower and lower in this value, at about 1 angstrom, you start to be able to visualize hydrogens. Right? So normally, if you look at structures in the protein data bank, many of them don't have hydrogen atoms. And maybe you've asked yourself, or you've seen this in the past, and you've asked yourself why. Well, the reason is because unless you have high enough resolution, hydrogens are really small. You're not going to be able to see them very easily if this is what your data looks like. Right? So imagine trying to detect a tiny little hydrogen atom sticking off here, or if this is a methyl, imagine trying to detect 3 little methyl protons sticking off of there. You're generally not going to be able to do it. X-rays are also sort of what you're measuring is scattering from electrons. So hydrogens only have 1 electron. I mean, they have 2 if you're covalently bonding and you're sharing them. But they only have 1 electron to deal with, and so that's only 1 electron to scatter the X-rays. And so you're generally not going to be able to see hydrogens unless you have a very good resolution structure. And then finally, there's some other stuff that the PDB tells you, as far as validation of the structure. They'll tell you if there's steric clash. They'll tell you if the Ramachandran plots looks like it should. So things like that. And so we'll look at specifically how these work. And then finally, we'll also see that many PDB files, particularly those determined by X-ray crystallography, have water molecules in them. And what happens is as you crystallize it, even though protein crystals look like little crystals, they actually have a ton of water in them. They're actually mostly water. And sometimes those water molecules are somewhat associated with the protein. And so they're not just exchanging and flopping about like a water molecule in solution would. And so you can actually sometimes see those water molecules in the crystal structure. On the other hand, again, remember this is what our raw data looks like. If I have a blob here, it's not quite this simple, but maybe that blob is actually a water molecule that's somewhat associated with my DNA. And so you can get to a point where if I'm just like, oh, I have extra density, I'm just going to add a whole bunch of water to sop that up. It can improve the agreement between the model and the experimental data, but you're basically just adding, it's like fudging your graph to make the fit look better. And so if you start adding too many water molecules, you should start getting a little bit suspect. And so I have some rates here. There's this value is sort of what I was trained on for structures that are about 2 angstroms or higher, or 2 angstroms or more. So about 0.2 to 0.8 waters per amino acid. That ends up being about 2 to 8 waters per kilodalton. If you're at higher resolution, though, you can generally do a little bit better. You actually are seeing real waters at high resolution. And so that goes up, and so you can get about 5 to 15 waters per kilodalton. If you're curious, there's a little bit of an analysis at this paper, and you can go look at the graph there. But for example, remember we talked about the PDB, everything given a 4-letter code. So 7B40, and I have no idea, this is just something that was submitted a couple of days ago. This has an R-free of about 0.19, so that's actually really good. A resolution of 1.25 angstroms, also a very good resolution. And there's 564 water molecules for 153 amino acids, but it's a tetramer in the structure. And so 612 amino acids total. So you can see we're right here in about this 1 point, or I'm sorry, 0.8, 0.9 water molecules per amino acid range. And the total molecular weight of that crystal, the tetramer, is about 65 kilodaltons. So we're very, very appropriate for what we're seeing here. Okay, and so let's take a look. So again, this is the PDB, but let's take a look at some structures or some entries in the PDB. And so if I go here to the PDB, again, generally if you go to the PDB, you often know the four-letter code that you're interested in. But we're going to look here at this guy, 3TJW. Oh no. Okay, there we go. And you can just search for that directly. And so on the website, this is the villain headpiece. So it's actually a very small sort of protein. I think it's only 40 or something residues. 34 residues. So you can see as you scroll down, they give you a sort of static picture of the protein. So this is fairly small. It's drawn out here. But in this page, in addition to the structural information, who deposited it, when was it deposited, those kinds of things, you can sort of see that there's some validation data, right? So this is the data that you want to look at to say, is this a good structure? So for example, here is where you would look to see what the resolution is. Here's the R-free value. Here's the R working value, or just the R value that we talked about. So again, notice that R-free is always higher than R work, or the R value. And this is sort of, I think, what you would get if you take the entire structure data set and look at it. And so it's actually really good agreement. They also give you this information over here. And so this is, every structure that gets uploaded into the protein data bank nowadays, there's some automatic checks that are run on it to make sure that everything looks good. And so, for example, this shows you, this is what the R-free value is, but it gives you the percentile relative to all the structures in the PDB, and it's doing pretty good here, right? So this is like probably top 10% of all structures. But then compared to other structures out of similar resolution, you can see it drops down a bit. So for all structures at 1.46 angstroms or better, this R-free value is probably in the upper 2/3 percentile, the upper 30%, or something like that, maybe 40%. Similarly here, compared to all structures, clash score is measuring steric clash. And so compared to all structures, it's doing okay. It's a little bit better than average. But compared to other structures of a similar resolution, it's probably in the bottom half, right? And that's not a reason to throw it out. All structures get curated, and so all structures are if you have too many clashes, they're going to say, hey, this is nonsense, refine this better. But it does give you a feeling for, okay, there might be 1 or 2 clashes in here that might be representative of real problems in the experimental data. But again, keep in mind that these are experimental conditions. Maybe there's something energetically going on that's actually causing there to be a steric clash, right? So if I put this in some kind of solvent, in steric clash, remember, you're just assuming that atoms are hard spheres and they're bumping into each other. But remember, in reality, atoms are actually surrounded by electron clouds which have some deformability. So if I put something in some ridiculous solvent, and Villen's actually a great example of this, maybe I can get the conformation of my protein to adopt something that's a little bit different than what it might adopt in normal aqueous solution. So just because you have 1 or 2 steric clashes, it doesn't mean that it's the end of the world. If I have a really, really strong electrostatic attraction between 2 side chains, maybe that's going to draw those 2 atoms together than you might normally see. And so don't be too scared about that. This one structure does really well in other areas as well, so Ramachandran outliers. So those are phi-psi angles, remember, that fall outside of the Ramachandran region, or the allowed region. There's no side chain outliers. And this is a real space refinement outlier Again, we're not going to get into the technical details, but again, this is kind of a local R value, and it's doing pretty good there too. And so going back over here into the slide, you can sort of see where this information is, and you know, I've just copied and pasted here too. But it's helpful to be able to see it in the context of the PDB. So I would say this is overall a really good protein structure. Let's take a look at another one, right? So this is staphylococcal nuclease. We don't have to go to the PDB because now you've seen where it is. So here, if I look at it, it's 1.65 angstroms. On the plus side, it doesn't really have any Ramachandran outliers, and so that's good. If you look here, though, the clash score is actually pretty poor compared to most other proteins of similar resolution. The side chain outliers, this is side chains that have an oddball conformation. Those are also doing pretty poor. And so if I'm really, really interested in everything, this is probably going to be an acceptable structure, but I might want to take care if I'm really trying to dive into specific regions of the side chains that I'm like, you know, suppose I'm mining the PDB and looking at side chain conformations. This one might have some oddballs that we might have to deal with or look at a little bit more closely. This is an older structure, so I guess I do have to, because I don't have this on my on my slide. So this is an older structure. It was published in 1990. And what's interesting here, if you look, there's no R-free, right? And that's because science is a process. When this structure was published, R-free really hadn't been popularized yet. And so not every structure you have is going to have every single potential validation statistic that you might be looking for. Some of this is easy enough that the PDB can just automatically go back and regenerate it. But remember, R-free requires going back to your original data, putting it in a lockbox, re-refining your structure, and generating everything again. They're not going to do that for the 30,000 people who submitted structures to the PDB before R-free became a thing. That would be kind of rude, in my opinion. And so they're not going to report it, but they're not going to calculate it themselves either, because that would be potentially doing something that the original investigators wouldn't approve of either. So just keep that in mind. You're not always going to see R or R-free. And as we did yesterday, yesterday we actually downloaded the file and took a look at it. You can also go in here and just view it. And so if we take a look at the PDB file in the web, again, we looked at some of this yesterday, but remember, there's a lot of remark information up here at the top. This is a lot of details on how the structure was determined, refinement conditions, software that they used to determine the structure. But then the main thing that you guys are going to care about are going to be these atom coordinates. And as Matt sort of highlighted yesterday, each of these columns means something. But it's something that you and I can also look at and interpret as well, right? And so, this is atom number 83. This is the carbonyl carbon, which has the IUPAC code C. It's an alanine residue. Some proteins, like if you're looking at a tetramer or an octamer, are going to have different chain identifiers. So you might go A, B, C, D, E, F, G. And so this is in chain A. There's occasionally some other data here. Like, for example, sometimes you see multiple conformations in the structure. And so maybe some of the molecules crystallize this way, or a side chain is crystallized in this way, and another other molecules crystallize in the other way. Maybe I should try, how about that? So one's this way, one's that way, and you'll see a mixture of both, right? And so sometimes the experimentalists say, well, I don't want to pick one, because the data have both conformations in there, and so they'll give them an A and a B conformation. And so sometimes you see multiple A's and B's here. And then these three columns are the X, Y, and Z coordinates. This is an occupancy code. So again, if it's a 50-50 mix, it'll say 50% of it's in this conformation, 50% of it's in this conformation. So that's your occupancy value. And then this last one is called a B, or a temperature factor, and that's telling you something about the motions. So again, these are atoms with electrons around them. that are, they're going to have some stochastic motions from like Brownian motion. And so if this number is large, if the B factor is large, you can see that these guys are going to be a little bit more motional than maybe something like this, which is going to have a little bit less motion. This is also someplace where you can sort of take this column and co-opt it for your own purposes. And when we get to the Python section, I think I have some code examples that allow you to sort of take this last column, and let's suppose you've measured a whole bunch of amino acid mutations doing mutagenesis, and you want to color all the mutations, a certain color. You can actually go into PyMOL and color by this B factor column, and if you've put your own number in this B factor, now you can color the structure based on some other numerical metric. And so that's a really nice trick that people do all the time. Okay, so we've looked at this a little bit, and we've skirted around the issue. It's time to actually dive in. And so there can be a couple of ways of obtaining PyMOL. I know Matt is a big fan on Chimera. I think Chris likes Chimera too. Chimera is actually legitimately free. And that's great. I think it's a really nice software to use. If your lab uses Chimera and they shun PyMOL, then you should use Chimera. Many of the principles that you'll see here as we talk about PyMOL are the same. So it's not going to be a waste of your time. On the other hand, I view it like PyMOL is $600 for an entire lab for 3 years. So everybody in my lab can have a licensed copy of PyMOL. And if my lab somehow grows to 15 people, that's still allowed. I don't ever see it growing that big. But and so for me, that's not a huge expense. On the other hand, you can also get PyMOL for educational use, and you can download it, like for this course, you can download a license, and strictly speaking, they're not like babysitting you. And so if you want to download a version of PyMOL from the PyMOL website and say that you're using it for a class, I'm not going to stop you. PyMOL might complain, and they might say, well, you're violating the terms of the license agreement, but again, they're not checking at this point. The other thing that you can do is if you're running Linux, or even if you have a Mac, you can actually install what's called open source PyMOL, which is basically all of PyMOL except a few little features that Schrodinger added to it. And it's perfectly functional, and you don't have to worry about the licensing at all. And again, since everybody in my lab has a Linux workstation that they sit at, this was the solution before I decided to bite the bullet and buy the licensed version. So there's some options here that we can do. You can go online and you can download it directly. And so in this case, if you go to the PyMOL educational site, you can enter this username and this password and download an educational license. I think I also put it on the Bootcamp website so that you don't even have to do that. So yeah, you can download the license here. You can download the actual software from this link. I promise you it's way easier to install than Cygwin. And basically when you first start it up, it'll ask you for a license file. You just point it at that license file and it'll work for I think it's like 6 months. So it's pretty generous. There's older versions of PyMOL which also work very easily, and they're very easy to install. So you can download them here, although these are going to take you to my server. So again, you're probably not going to be able to use Chrome for that. But it's fairly easy to get a copy of PyMOL, and it'll work. The educational license is not restricted, so you can pretty much do anything you need to do with the educational license. And so even if you're not in a lab that's purchased it, you should be able to get by on that. You just have to download the new license file every few months. On the other hand, if you want to install the open source PyMOL, so PyMOL started out as open source, its initial license was open source, it was written by a guy named Warren Delano, and he had a vision of it being open source, and then Schrodinger sort of took over, and that's why we have this situation that we're in now. Keep in mind, that could happen to Chimera. I mean, nobody envisioned PyMOL going to closed source or being proprietary when Warren first wrote it. But you can still get the open source values. And so if you can get apt, or if you have administrative access on a Linux system, you can install PyMOL that way. That's one way to do it. On CentOS, it's a little bit harder. And so this should walk you through it. We're not going to do this here because that would probably turn into a fiasco like installing Cygwin. But I've done this. If you have questions and you try this on your own system or you try it on a system, I can probably give a little bit of support. But honestly, this is one of the reasons why I like to run Ubuntu or Debian, because it's really just a matter of typing one single command and now PyMOL is installed with no licensing restrictions whatsoever. There's alternatives. As I've mentioned, UCSF Chimera is one of them. It's absolutely free. There's no licensing problems with it whatsoever. It's a little bit trickier to use. And so particularly for students in my group, I would prefer that they sort of focus more on the structure and the molecular side of things than having to learn a whole bunch of syntax, and the user interface I don't think is quite as polished, but it's overcomable. I mean, certainly there's a ton of people who use it. So I would say if you're in doubt, download both and see what you like a little bit better. VMD is great for looking at molecular dynamics simulations. So if any of you guys are in a computational lab, you'll probably be using VMD for this. PyMOL is optimized for like one structure at a time, but in molecular dynamics simulations, you're often looking at thousands of structures. So VMD doesn't have nearly as nice of an interface as PyMOL, but VMD looks at many, many structures simultaneously, and so it's really good for that. If you're curious about the history, I mean, I kind of like to geek out on this kind of stuff, and so you can follow that link. A lot of the technology that's in our, that Pixar uses, that that sort of goes into 3D televisions and stuff like that, actually had its first appearance for these kinds of advanced scientific applications like molecular visualization. And so this is a case where many of the technological advances can trace their way back to visualizing or computer-aided visualization or design. And that's a little bit represented here in this history site. Okay, so hopefully at this point, and we can give you a couple minutes if you need it, but hopefully at this point you've had a chance to at least download PyMOL. You can download the license off of the website, get it running. It's really not going to take that long. And in this case, once you start it, you'll see a window like this. Now, if you're running the educational PyMOL, you'll see like an EduPyMOL banner here. And so that's not going to be a problem. It's just showing that, hey, you're using the educational version. As soon as I think you click into the site, everything goes away. We do have on the website this PyMOL tutorial. And this is something that I've written many years ago and I've kept updated along the way. And so this is kind of how I would recommend you guys, if you've never used PyMOL and you feel like you're lost after this particular session, this would be a great thing to sort of read and work through. There's examples. You can sort of ask questions. This year I totally rewrote it, and so you're going to be looking a little bit at SARS-CoV-2 proteins. And so hopefully that makes it a little bit more relevant. But it sort of walks you through the main features. And then additional materials that we're not going to get to at all today. We have a tutorial on, a reference that was written that maybe goes into a little bit more depth on using PyMOL commands. And so there's all sorts of, this is rather dense, but it's a handy reference sheet that you might find helpful. And other stuff on the website is, I think there's an additional, no, that's pretty much all we got. So between those three documents, you should be able to, between the slides, the tutorial, and the quick reference, you should have a pretty good handle of things. The tutorial often also links you to a couple of other online resources that you might find useful. But anyway, so this is PyMOL, and you might say, well, where's the molecular visualization? Well, that can be done a couple of different ways, right? So one way to do this would be to go to the PDB and select download files, and I can download in PDB format. And not exactly sure where it put it. Okay, it's not where I would have put it, but we'll drag it over here on the desktop. And so you can either double-click this once PyMOL is installed and it'll open it up, or you can go here to File, Open, and navigate to the PDB file. All well and good, very, very straightforward way, very similar to how you've done other structures. On the other hand, again, if you're just starting from blank, you can, at the PyMOL command line, so again, notice that you have a command line up here and a command line down here. Typing into one doesn't do anything, or doesn't affect the other, but you can type into either. So if I type fetch 1SNC down here at the bottom command line, you see it brings up the same structure. So that's a very easy way to get a PDB entry from the protein data bank. If I go up here, so I'm going to delete this structure, or delete that. If I go up here and type it into the upper command line, fetch 1SNC, it's the exact same structure, it's just a different color. So doesn't make any difference whether you type commands up here in the command line or down here in the command line. The real advantage up here is that you can sort of cut and paste and copy and paste. So if I type, something in here, like let's type fetch 2OED, so that's GB3. Up here, if I drag and select, I can copy and paste that. Whereas if I go down here and I start typing, I can't drag and copy and paste it. And so up here is kind of where you can sort of use it as a conventional text box. If you type down here, it does the exact same thing, but it's not going to let you sort of drag the mouse over and copy and paste. You can use your arrow keys to move things around so you can sort of see. Again, notice as I'm moving things down here, the cursor is not moving up here. So if I move the cursor left and right, nothing happens up here. That's perfectly normal. Those two are not synchronized. This is sort of the main graphical display. And so again, this is 1SNC. You can see the protein is rendered as this cartoon model that we've been sort of showing you throughout the sessions. You might ask yourself, well, what are these little crosses and these little dots and things? So these little red dots, those are the water molecules. Those are called crystallographic waters. If you don't really care about the crystal structure and how it's determined, you can actually very easily remove those. PyMOL also includes other atoms. In this case, these are heteroatoms. And so this protein is involved in binding nucleotides. And so there's a nucleotide analog here that it's rendered in stick form, just like your organic chemistry classes have. And then this green sphere, I think, is a calcium ion. And so it's chosen to render things that aren't proteins using a more conventional chemistry approach. And so I can click and drag to move things around. I can right-click and zoom in and zoom out. So, and then if I use my middle mouse button, if I hold down the middle mouse button, I can just shift it to the left and right, up and down. Now, many of you will have a scroll wheel on your mouse, and that actually has a function here too. And so if I scroll my wheel down, so I'm moving it down towards me, what's happening is I'm basically adjusting the slab, right? And so slab is something that's a little bit hard to define. I have an example here in the in the bootcamp tutorial, where you can think of SLAB as sort of selecting the window of atoms that you're viewing. So if this is you over here, and you're looking on the molecule this way, if you adjust the slab and you make the slab small, you're basically cutting out or cutting away the ends or the extents along that sort of visible axis. And so you can see here, I've basically cut so that I'm basically looking at a very narrow window through the protein. And if I push my mouse ball away or my mouse wheel away from me, you can see I'm basically allowing me to view the entire protein structure. So let's dial it back down again. So if I dial my slab down so it's really small, now you can see I'm moving it, but only a tiny little fraction of molecules are cut through. And you can see down here, here is where this helix is starting to intersect my slab, and I'm not seeing anything that's coming in front of that front plane. And so that's useful. That's useful if I want to look at the middle of a structure. So here I'm going to render everything in a space-filling model. And let's suppose I want to see something at the active site. Well, now I can adjust my slab so I can cut away yeah, there we go. So what I'm doing is I'm basically cutting away, and now I'm viewing basically into the guts of the protein. And so I've basically drawn a plane in front of which no more protein atoms are going to appear. And so I can look and say, oh, look at that residue is doing that. Whereas if I'm looking at the whole protein molecule, I'm never going to be able to see into the middle of the protein. And so the slab gives you a control for selecting atoms and hiding them that are basically you don't want to see or that are going to interfere with what you're looking at. Okay. Other things in PyMOL that we can talk about in terms of the basic user interface. First of all, note that I have a couple of different windows. So first up here, this region is kind of where a history of all the commands that you're typing are. It's scrollable, but you can see that, if I showed spheres, it's going to show me that I typed that. And if I click on some other things, like suppose I click on this atom, it's showing me that I clicked on an HOH atom. It's HOH number 212. I clicked on the oxygen. Again, you can't see hydrogens here, so it's only showing you the oxygen. And in this case, if I click on different amino acids, you can see that I can say, oh, this is what my selection is. Now, right now, it's adding to that selection. And so if I just click here without anything else, it says it's a selection defined with one atom. But if I click on an amino acid, it's going to select the whole amino acid, right? And so here, I clicked on C delta of lysine 70, but it's selected all 10 atoms in that amino acid. So this is where sort of your command history is shown. This is actually really helpful if you're keeping a lab notebook to copy and paste. If there are ways in PyMOL to sort of log what commands you're typing, but those I don't find them to be particularly helpful. I find it much more helpful if I'm trying to sort of keep a track of what I'm doing, is at the end of my session basically select all of this, and I can right-click it and copy it and paste it in a text file. And now I have a command history of everything I typed in that session. And if I want to go back and recreate that image, I can also cut out from that text file the things that I don't want to put in there. Or I can share that with a colleague and say, this is how you should make this image. Right? So there's some tricks here with PyMOL, but it does give you access to that command history that you can go cut and paste later on. Over here are some options on how to display the molecule, and so you can adjust different zoom levels. So you can zoom the whole molecule. You can see it sort of zooms in. This laptop is a little bit aged, and so it doesn't necessarily have the most high-performance graphics that we're dealing with. We'll get here into X-ray or ray tracing, but the other thing is that you can do a whole bunch of stuff with PyMOL and to make movies. And so we're not going to cover movies or molecular movies at all, but a lot of these sort of buttons are what you would do is if you're trying to adjust movies or move frames in a movie. You can also go in and actually build in atoms or conformations that you might want to do to test things out. Again, that's a little bit beyond what we can cover here in this period. So the other area in PyMOL that you might care about is this area over here. And this is basically a list of loaded objects. And so right now I have one protein structure loaded. That's one SNC. If I click on that, I can make that entire object disappear. PyMOL also has selection objects. And so remember I told you if you clicked on something, you're selecting a certain number of atoms. And so here you can see these atoms are what's being selected. I think it's also grabbing that water molecule over there. But I can hide that as well by clicking on it. And then for each, and then if I click on all, it toggles everything. But then for each object, I can go in here and there's 5 different buttons. So one of these, the one that has the rainbow picture on it, is basically allowing you to color different parts of the atom. And so suppose I want this whole protein to be colored red. Now that looks horrible to me, but it certainly did what I told it to do, right? But there's also other options, like I can color by secondary structure, and it gives you some options for how that would look. And so you can see here where at this point, when I'm looking in all atoms, it's really hard to tell whether this is an alpha helix, beta strand, or coil. I know that most of this structure is going to be beta, and so in particular there's going to be a big beta barrel here, and then a couple of alpha helices on either side. And if you were to talk to Bertrand Garcia Moreno, he'd say that this has a definite correct secondary or a definite correct orientation. I think this would be it. But anyway, so here you can see where it's colored by secondary structure. And then if I want to show that cartoon, right, so that's where this S and H. So S stands for show, H stands for hide. And so here, let's suppose I can hide my spheres. And now I'm looking at secondary structure again. And so you can see that the secondary structure basically shows that there's this yellow beta barrel, there's these loops, and then there's these red helices that come down and form the back end, I guess, of the enzyme. And so coloring is useful. You can go into different representations, and so you can play with all this. Again, cartoon is basically what we're looking at here. If I show spheres, again, that's showing your space filling. Again, if I hide those spheres, I can hide the cartoon representation, and now I'm basically only looking at the little dots represented by the water molecules in this analog here. I can also show sticks. There we go. And so you can see this is showing the bonded sticks. So there's a couple of different representations. Each has their own advantage and disadvantage, right? So this is maybe a little bit more chemically accurate than the cartoon model, but it's a heck of a lot harder to understand or to grasp. Like I said, if I look here, if these weren't colored yellow and red, I'd have no way to say where's the helix, where's the strand, what's the secondary structure of this protein. On the other hand, if I'm interested in where the atoms actually are, if I hide this, well, let's show the cartoon, and we'll hide the sticks. This doesn't really show where atoms are at all, except the backbone, right? So if I'm interested in side chain structure, the cartoon doesn't really get me anything. Okay, so now we've sort of covered basic visualization. We've covered the different types of representation. Here, this L is for labeling. And so if you want to label things, although I rarely use this, actually I rarely use this mouse region at all. But if you want to label things with like one-letter codes, you can sort of show that here. And that's one way to do that. Clear the labels. And then down here, this is kind of your mouse functions. And so as I said earlier, if I use my left mouse button, it's giving me rotate. Middle mouse button is move. Right mouse button is move in the Z direction. And then wheel is slab. If I hold shift, I can start selecting atoms based on where I'm clicking. And so that's something that can be useful. But it's telling you all that here. So for example, shift gives me a box that adds to my selection. Shift with the middle button gives me a box that subtracts from my selection. So you can see that makes that selection go away. I can adjust all sorts of other stuff, like I can pick individual atoms, I can pick residues. Again, some of this gets to be a little bit pedantic, and you're not going to need to know all of this. Honestly, most of the time between Control and Shift and just using the mouse without any of those modifiers, I can do pretty much everything I need to know. So I'm not often using Control Shift or double clicks or single clicks. You can also go down here to click on some of this. So if I click on selecting, you can see I can control whether I select residues, chains, segments, objects, whole molecules. Again, just C alphas. So now I'm only selecting one atom at a time. And there's other options here too. So if I click on the mouse mode, I can go into other modes. Again, 3-button viewing is probably what you want to hang out in most of all. But sometimes you only got a 2-button mouse, and in which case that'll show a 2-button editing or a 2-button viewing. That's even more complicated to get all the features that you would want. Okay, let's go back to 3-button viewing, and we'll be selecting residues. Questions so far? Okay, so let's move on to the command line interface of PyMOL, because honestly, the command line makes this so much faster, right? I can go in here and I can click things, and I can I can drag to select different residues, and I can color those residues. That's one way to do it, and if you're really scared of the keyboard, that's a great way to do it. You can get done almost everything without using the mouse at all. It's just going to take you some work. Most of the time, you're going to be working at the command line if you want to stay sane. And so in this case, there's a whole bunch of commands. And some of these are outlined in the PyMOL tutorial, so you'll be able to go through them and sort of learn the commands as you go. But we'll just highlight a few of them here. So the one that we can talk about that we've already seen, the mouse interface, is color. And so, for example, if I type color red, now I color my entire molecule or everything in the screen red, right? Now that's not so useful because, again, it's very rare that I want to color everything the same color, right? I've lost all of the information, the chemical information, in terms of some atoms are colored green, like carbons are colored green, nitrogens are blue, oxygens are red. When I color something red, that can be great if I'm trying to differentiate between entire chains, but I lose a lot of information this way. So for example, if I fetch 2OED, now this is a totally different structure, so there's no reason to believe that they should be steric clashes or whatever, right? These are just two different molecules, one where I've loaded staph nuclease and one where I've loaded GB3. You can see that, okay, one of those is colored red, one of those is colored yellow, and so that makes it easy for me to distinguish between one and the other, but it's not going to let me distinguish a carbon atom from a nitrogen atom, for example. So let's load something a little bit different. So I'm going to delete both of these. So I can go here to Action, and I can delete. So there's this first character, I guess, contains actions. I can delete objects. And so now I have nothing again. I can probably delete this selection. Okay, let's fetch, I don't know, what's a good multi-domain protein? Well, I guess we'll move along in the slides, so that way at least we'll have the structures loaded when we want to talk about them. So we're going to load a protein called PIN1. This is 1F8A. And so you can see here it's in pink. Again, the subsequent structures that you fetch are given different colors for the carbon atom. If I show spheres, though, you can see that carbons are pink, oxygens are blue, red, nitrogens are going to be blue. So it's just the carbons that get the different colors. If I go in here and I go to my action, you'll see that there's a button called Remove Waters. Very useful if you don't care about the waters. And so now all those waters are gone, and I'm basically just left with protein. And if I look here, and I go back to my color example, right? So I can color everything yellow, but oftentimes I only want to color some part of the molecule yellow. And so this is where the selection syntax comes in for PyMOL. And so PyMOL has a whole bunch of features that allow you to select specific elements or specific parts of your structure. And so in this case, if I want to select only carbons, I can put a comma here and say color yellow, comma, LMC, which means all carbon elements. Notice that's different from, say, and again, I'm just going to type it up here just to show you that. Let's do color green, and let's type name C. So notice that not every carbon was colored green, but some were. Right? So when I use the name selection tool, I'm selecting atoms that have that specific name. Remember we went into the PDB file and we saw that some atoms were named CA, some atoms were named C. So name allows me to select atoms by name. LMs allows me to select them by their chemical element symbol. I can also go and select entire residues. And so if I type, I just clicked on residue 83, and this residue is 70. So let's color red, and I'm going to type resi 70 to 83. And so now I've selected, or I've colored red, but I've only done it for the residue index from 70 to residue 83. Right? So resi allows me to select residues by index. As you might imagine, there's another one that allows me to select residues by name. So color green, let's do res N, and we'll type gly. So it always uses the 3-letter codes. And this colors all glycine residues green. Right? And so I can build these selections up. This is like any other search tool. Like if I go to Google or PubMed or whatever. And so let's reset things. One useful utility or one useful trick is to type util cbag. And this basically colors the atoms by the residue or by the element type. And the last letter gives you a hint at what color carbons will be. So if I type util cbag, I basically go back to my default coloring scheme where all carbons are green. If I type util.cbay, all carbons are going to be yellow. But nitrogens are going to be blue, oxygens are going to be red, and so forth. So it's a helpful tool if you've really screwed up your coloring and you want to go back to something that's sensible. You can use that util.cbag or util.cbay. Okay. So let's go and do something a little bit more advanced. So let's say I'm going to color red, and I'm going to color all glycines, so res name glycine. And I want to only select glycines between residue, 10 to 75. So that'll be the first domain of my protein. So I'll do residue glycine and residue index 10. Actually, I think for this protein it's probably 55. And so now you can see I've selected or I've colored those atoms red within a particular residue range and that have a particular residue name. And so let's go and, again, we'll keep it simple for now. I'm going to hide the spheres. And so you can see there's only 2 glycines in that first domain that it colored red. What if I want to show them as a different, this is a great way to highlight things, but oftentimes if you're in the back, you probably can't see which ones are red. So PyMOL allows me to do all of those visualization types as command lines as well. And so in this case, I would type show spheres. And I can type the whole thing, res n gly and resi 10 to 55. And so now I'm showing only the spheres of those glycines, and so you can sort of visualize where the glycines are a little bit easier. And so show has a lot of different options. Let's suppose I want to do show sticks. And we'll do the same kind of thing. We'll do resi 10 to 55. And this time we'll label, anyway, give me your favorite amino acid. Tyrosine. Okay, res name tyre. And so now I've shown those residues as sticks that are tyrosines between residues 10 and 55. And so you can see them there, and they stand out too. So this is a way to highlight residues in a representation that maybe isn't quite ideal for highlight individual atom positions. Now you might say, well, that's great, but do I have to memorize all this stuff? Because that would be hard. I already have to memorize too much as it is. Well, if I'm in this window and I press escape, notice that the graphics go away and I'm basically left with another window of text. And so from here, I can type help show, and that actually gives me some information on what I can show. And so here, I can see all of the representations, so lines, spheres, mesh, ribbon, cartoon, sticks, dots, surface, etc. I don't even know what all these do. And then it gives you some ideas, or some examples, like show ribbon. But then it shows you that, you can put a comma here and select a specific atom. If I'm interested in those selection terms, I can type help select. And that gives me an overview of the select command. But if I really want a detail on how to form selection strings, I can type help selection. And now I see all of the different options that are available. So I can select them by atom name, residue index, chain ID, segment identification, element, so element type, all of these things that go in here. And then there's other options to select things by distance. So I can select distances, like anything within 10 angstroms of this other atom that I'm interested in. I can select, by B factor. I can select all of the things that I might be interested in selecting on. And again, the world is your oyster. You can do whatever you want in terms of selection strings. Okay, questions on this so far? So you guys have the tutorial, and raise your hand. I feel you guys can interrupt me at any time. You have the tutorial available to you. It overviews a lot of this stuff. The remaining time I'm going to spend going into some more advanced stuff that you might not see in the tutorial, but that might be super useful if you're making images. And so, again, keep in mind, you might have a protein, you want to put a pretty picture of your protein on your poster at the end of the summer. This is one way to do that. And so we'll start looking at how to do some of these things. So one thing that you can do is if you have two similar structures, you can go in and you can load them both. So we're going to load up another structure of pin 1, one pin. And I'm going to go in here, and my default is always just to delete waters, because I rarely care about those. And if you look here, this is similar structure, but it's not really easy to tell that it's the same thing. And so PyMOL gives you a command called align, which allows you to align structures that have similar three-dimensional or tertiary structure. And it uses some bioinformatics tools and structure alignment. We'll talk about that tomorrow. But basically, I can do align one F8A and then comma one pin. And it's going to give a specific alignment based on those residue types. And so it looks at residue similarities, it looks at structure similarities, and says, based on that, I'm going to move this second molecular object so that it matches as closely as I can possibly get it to this first molecular object. And so that's what it's done. And now you can see quite clearly that these structures are by and large extremely similar. Now notice there's one region where they're not similar at all, right? And that's this yellow loop. And having worked on this protein, there was a big debate in the field whether this yellow loop was maybe some kind of gate that closed and opened over the active site and maybe controlled the function of this enzyme. And eventually, enough structures were determined and experiments were done, and people realized that this is basically moving, or this is basically a section of the protein that's actually interacting with the next crystallographic unit cell. And so remember, these are crystal structures. So I have a molecule here, a molecule here, a molecule here, a molecule here. PyMOL only shows you one. And in this case, even though nothing is drawn over here, this would be interacting with the next pin 1 molecule in the unit cell. So it would be like this thing, where a loop for 1 is basically coming over and interacting with the other unit cell. But that's a useful tool if you've got multiple structures. And again, if I want to color one, let's say I want to color one pink, and I type 1F8A, now I have one pink and one is kind of a bluish gray. We can also go and measure distances. And let's suppose I want to see, what's the distance between this tyrosine and this glycine here. Well, again, I can go up here to Wizard, Measurement. It defaults to distances, but if I click distances, you can see you can measure a whole bunch of different things. And what I'm basically going to do, it's asking me, please click on the first atom. And so I'll click on this glycine residue. It looks like I clicked on the carbonyl oxygen. And now I basically have to find the right orientation. But you can see when I click on that tyrosine, it now gives me the distance as 10.4 angstroms. And so that's a very useful tool that you can go in and measure distances. You can also go in and measure dihedral angles, right? And so if I wanted to oops, forgot my comma. Okay, so here is the backbone of the 1F8A structure. I'm going to go utils cbay because I want to see what my atoms are. Okay. So remember, if I have C prime, N, C alpha, C prime, those are the four atoms that define phi. And so I can go here to measure dihedrals. And if I go C prime, N, C alpha, C prime, you can see it's giving me a dihedral angle of 135 degrees. So that's one way that you can actually go in and measure those Ramachandran angles that we talked about on the first day. One neat trick that you can do is this ball and stick effect. So this kind of makes it look like your organic chemistry classes. So right now I have, let's hide all this, and let's show sticks, and we'll look for residue. Let's do 70 to 80 again. We'll do center visible to bring that to the middle of my screen. And so normally if I type now show spheres, and if I type visible, that's going to select everything that's currently visible. Normally those spheres are space-filling spheres, and so they take up most of the space. I don't get this effect. But PyMOL allows me to set the sphere scale so that I can scale that down and get something that looks a little bit more like a toy model. So if I go here and type set sphere scale equals 0.3, and I don't have a typo. Oh, I think this might be, so I probably need to change this. It used to let you do an equal sign. I think you need a comma, yeah. So now if I use, so note that for all of these, this is going to be, I'll probably update this presentation. Hadn't even realized that. So if you look here, now those spheres are nice and small, and it gives you that kind of ball and stick type of representation. Now let's suppose this was my protein and I wanted to make this really nice looking for a poster. Right? I can go in here and do, go to display and change my background to white. Again, your poster is probably going to have a white background or something similar. And if I go in and now go up to this ray button, this is this button that we talked about earlier, that's going to give us what are called ray traced graphics, which are a little bit higher quality than what PyMOL is able to render on the fly. And so here you can see I can select how many pixels high and wide. I can also go in and sort of calculate how many centimeters are. Remember, our good quality graphics standards apply here. So generally for a poster, you want to go shoot for 300 dots per inch. But think about how big your graphic is going to be. You probably don't need to do a billboard size. For here, we're just going to click ray just to see what happens to it. And so you can see this image has now become a lot higher quality, right? And I'll just click in here so you can see that there's some graininess around the edges, and not all the lines are smooth. If I go up and hit draw and then go and hit ray, not only does the background disappear, so I can now drag this onto a colored background and it should look decent. But the image quality, there's shadows, there's a lot more smooth lines here. And so this is going to be what you want to do if you want to make a fancy image for your poster. And once you do that, you can actually save it to a file or copy it to the clipboard. It's your choice. I usually recommend saving it because I think then you have a hard copy. But beware, if you click back into the visualization window, it goes back to the standard sort of conventional graphics that you might view that aren't quite as high quality. The other thing that you generally want to do is make sure your slab is pretty far out when you do a ray trace. Otherwise, you can get some ghost atoms where they're kind of transparent, but not exactly. I think that's a bug in PyMOL. But you can see this works really well. If I go show sticks, one FA and not visible, and actually I don't want to do that. I'm going to show cartoon. Right, so you can sort of see here how all this fits together. And if I type ray or I go up there and click ray, you can see it gives a pretty reasonable representation of the helices and cartoons as well. So it's not just these atoms, balls, and sticks that it's doing. There's some other things that you can do that can be helpful. So one thing that you can do is make transparent surfaces. So if I show surface, you can see what that surface looks like. And sometimes you want to go underneath and say, oh, can I see the atoms underneath? And so I can set transparency 0.8. And you can see that that's made the surface transparent. So now I can see what the surface of the molecule looks like, but I can also visualize what's going on underneath. And again, when I ray trace that, it can look really, really nice. Now, except it takes a while, particularly on this ancient computer. Okay, I'll let that crank through. Uh-oh. I might have asked it to do too much. Oh, there you go. So you can see, again, the surface actually looks really nice. It's smoothly rendered. And one way to do this is actually, I'm going to delete this. So I'm going to go in here and duplicate this object. Copy to new. And so I'll call this surf. Okay. And hide sticks. Hide, we'll just hide everything. Show surface object 01. And so that's going to be our surface. I can do show cartoon surf. And if I want to color them differently, so if I want to do color gray surf. So now you can see that the surface has one set of colors and the underlying cartoon representation has a different set of colors. And again, you can use this to highlight the surface but also show the structure. And that's kind of what's going on here. And then there's some tools that if you're working with nucleic acids, there's a lot of tips there. Again, I don't think time allows us to go through all of that, but these web pages, and then this is kind of my standard go-to approach for getting a reasonable DNA structure. So you can load up the Dickerson dodecamer, which is just 12 bases in a double helix. You can type these commands. And again, I think you can't use equal signs anymore. You have to use the commas. So I'll fix that and re-upload the slides. But once you do that, you can get to a point where you'll get this type of a structure, which actually looks pretty reasonable. And then there's the people that work with nucleic acids all the time, they have their own tips and tricks. And so you can go to their websites and try it out for yourself. And then finally, there's some ray tracing tips if you want to make your ray traces look as good as possible. Again, this ray tracing panel is actually super helpful because up until this, it was really unclear, how many pixels your image would have and how much pixel density you would have. And so you can go in and set your background to white. This lets you do a transparent background so that you can paste on a different fancy color. And then you can also go in and control how many dots per inch that you've got. And so that's pretty much what I have for PyMOL. It's not a hard program to use. I think there's a lot of cool things that you can do with it once you're familiar with it. Like most other things, you have to spend some time playing with it. There's that word play again. And but if you use this to generate models, PyMOL can be useful in both, showing and doing some analysis of protein structures. For example, is a residue exposed? Is a residue buried? It can also be useful in making pretty pictures and conveying those ideas to others. But always keep in the back of your mind that these are experimental models, right? They're not perfect. One of my old colleagues used to say, seeing is deceiving. And so you can make a model in PyMOL that has no accurate representation of what the real chemistry is, just because you've selected some atoms to be visible and others not to be, and maybe oriented in a particular way. So always try to represent the good chemical principles that you know when making figures like these. Okay, I think that's all I got to say for this. Questions on the PyMOL? Yeah, you guys are getting tired. I'm getting tired too. Actually, yesterday I was spent. I went home and I just kind of crashed. But today is a little bit easier because I wasn't talking as much. But I can only imagine, again, like I said, drinking from a fire hose, it gets old after a while. The advantage is that you'll be able to go home, and we're not going anywhere. So if you have questions, you can still email us, even after the sessions are over.